So we were discussing about the signal amplification. It means that whenever there is there is a signal transduction, the signal amplification is extremely important process, because the signaling intermediate persists long enough to stimulate the production of many molecules required for the next step in the cascade. So that's why the multiplication of the effects of a single receptor ligand interaction of the cell surface is huge. Just a single molecule of a ligand can give huge responses, because because of the exponential increase in the subsequent molecules, the exponential increase in the number of subsequent molecules. So I will give you one example in the next slide. Here you see just one molecule of the ligand is going to act with the receptor in the G protein coupled receptors. For example, when the glucose is needed by our cells, here you see just one molecule is there of the receptor, and after that you see the cascades. You see the inactive G protein, then the active G protein, and after that the active cyclic AMP, then the active protein kinase, and then these are actually the intermediate steps, right? And these intermediate steps, nature has given so many intermediate steps, and I'm gonna ask you why nature has so many intermediate steps during a signal transduction. I will get back to you to have an answer from you guys, okay? And at the end of the day, you see the response. The final response is that just using one molecule, the signal is amplified 10 raised to the power 8 molecules, right? So this is how the things are going on in the signal transduction. And also another concept you should see, that each and everything in the cell is present in ready-made form, just they need a signal to get themselves activated. For example, you went to a mall, okay, and you want to buy some shirt or trousers, and you bought it, but you just need, you can immediately use it, but before that you just need one step. Okay, that one step is to pay for that particular shirt or trouser, and once you pay it, it is ready to use. Similarly, that signal, that is the ready-made signal, is that you just pay and use it. Here you just have to get activated stuff. Inactivated stuff, ready-made stuff are present already in the cell. They are floating around in the cytoplasm, they are roaming around in the cytoplasm, they are waiting just for the signal, and signal is here. When the ligand is bound with the receptor, signal is there, and that signal is then transduced into the cell, and it will initiate all these cascade. One activated thing is going to activate the other thing, the other thing is going to activate the third thing, the third thing is going to activate the fourth one, and similarly till the response is reached, there is a huge amplification in the signal. So this is the concept of signal amplification. Okay, so one type of the receptors, it is G protein linked receptors, that they are also called as G protein coupled receptors, and they are activated in almost 40% of our receptors that are responsible for the drugs to act on the G protein coupled receptor. Like for example, if human beings are taking drugs or medicines, 40% of the medicines use a G protein coupled receptors to and then they get that response from the cells, right? So most of them, they are the hormone receptors, and the receptors that are responsible for our sense of smell. Okay, so why they are called a G protein? They have a guanine. This is a nucleotide binding protein, and guanine is rich in these proteins. Okay, that's why we call it as G protein coupled receptors. So they are very important as far as physiology is concerned, as far as they are very diverse. They have 40% targets, as I told you already, and they are only found in eukaryotes. They are not present in the prokaryotes. So here is the structure of the brief structure, or the cartoon of the G protein coupled receptor. This is the extracellular membrane, right? The plasma membrane here, and this one the extracellular part. This is the extracellular part, and here there is a G protein, right? Alpha, beta, and gamma. It has three subunits: alpha, beta, and gamma. This is a G protein coupled receptor. This is present towards the intracellular portion of the cell membrane. This is G protein, and this is the receptors. Why we call them as G protein linked receptor? Because they have a link with the G protein here. This is the G protein, and this is the receptor. That's why we call them as G protein linked receptors. These are also known as 7 transmembrane receptors, because they had 7 times they cross the cell membrane. Okay, and here again this is an image from the book, right? And these are the 7 helices: 1 2 3 4 5 6 7. 7 times they are going to cross the cell membrane, and then and of course this is a protein, right? I told you that most of the receptors they are protein in nature. Here we have amino end, and here we have the carboxylic end. And you know, the protein has lots of different domains, as we studied in the chapter 3, that the proteins, they have domains, right? So some different domains are present here. You see G protein coupled receptors, kinase, phosphorylation sites are present, then some other protein kinase A phosphorylation sites are present, then the messenger binding sites are present. So these are actually the different domains on this protein molecule. They are going to provide different functions in the signal transduction, and we will see how in the coming slides. So this is these are the steps involved. Here you see these are the receptors. This one, the single receptor here, and this is a G protein. An inactivated G protein is always having a binding with the GTP, diphosphate, guanine diphosphate. Okay, and once the ligand is there, the signal is transduced inside the cell, and when signal is transduced inside the cell, G protein is going to get GTP, and GTP is removed. So this triphosphate activated G protein, it will the alpha subunit will dissociate as soon as the GTP is attached. Beta subunit and gamma subunit, they are not as important as alpha subunit. So alpha subunit will take this GTP and is going to activate or inhibit some of the target proteins, initiating the signal transduction events. So after that, once the function is performed, the alpha subunit hydrolyzes, and its bound GTP is converted again into GTP, right? So this was all about the G protein coupled receptors, and that how they are going to perform their functions. Okay. So two important signal pathways. One is the CAMP, cyclic AMP, ADP, ATP. This is the youngest sister of ATP. ATP, adenosine triphosphate, then adenosine diphosphate, ADP, and then adenosine monophosphate. So that is C stands for cyclic adenosine monophosphate dependent pathway, and the other pathway is the IP3DAG, inositol triphosphate and diacylglycerol pathway. So these two pathways are involved with the GTP, with the G protein coupled receptors. Okay. So here we see the cyclic AMP dependent pathway. It will the GS or GI. I will tell you later that what is GS and what is GI. GS is stimulation, activation of any event, and GI is inhibition, the stopping of an event. Okay, so the ATP will be converted into cyclic AMP, and then they will activate the protein kinase A enzyme. So this pKa, when this pKa is activated, this pKa will perform the functions required, and it will create the response here.